Hello friends. So to start with the next lecture, that is RNA extraction. So the question is, why do we need to extract RNA? So RNA is extracted for research purpose, for diagnostic purpose, and in research we just want to see the gene expression analysis. We want to compare the expression of any gene, target gene, in particular type of tissues or particular type of disease condition, and even we are extracting RNA for diagnostic purpose also. So there are downstream applications for RNA extraction, like if we want to do some real-time PCR expression for gene expression studies, if you want to see the any differential expression analysis, if we want to do we want to compare two gene the expression of a particular gene in control versus disease condition. So for RNA extraction, for the first step is what are the different type of samples which we need to know that we can extract RNA from those samples. The first is we can extract RNA from whole blood, or we can extract RNA from any particular type of tissues. We can extract RNA from cell culture. Any type of cell culture you are doing and you are performing experiments, so from those cells, culture cells, you can extract RNA. Then you can extract RNA from the body fluids which are like other than your blood. Body fluid like throat swab, nasopharyngeal swab samples, nasal swabs, oropharyngeal swab samples, any other fluid which we can use from, even from bowel sample. So like that you can extract RNA from all these four different type of tissue samples. So these are the type of tissues. Now the second question is, what are the method which we can employ to extract RNA? So the first method is manual method, in which you can extract your RNA manually, and the second method is kit based method, where you can use some commercially available kits to extract the RNA. And the third method is if your combined method. That means if you are using your manual method combined with your kit based method. So there are lots of papers where they have used combined method also. Initially they have started manually and then they have progressed to kit based extractions to get a purified amount of samples. So there are some samples where you get lots of contaminants which inhibit your PCR, or which you are not getting clear RNA solutions, eluted solutions. So there you have to use kits for the purification. So these are the three different methods. So we'll go one by one to all three type of method for RNA extraction, because RNA extraction is tricky and it is very sensitive method. So first we'll go with the kit based, because it is easy and you can get RNA, good quality of RNA within 1 or 1.5 an hour. Manual extraction require more time to extract RNA, but it has many advantages over your kit based. Even kit based has advantage that you will not get too much contaminants with the kit based. But all these two methods, manual as well as kit based method, they have their own advantages and disadvantages. You have to see your application, what further application you want to use after RNA extraction. So based on that you need to opt your method, right? So to start with this, to start with this kit based method, kit based RNA extraction, all companies which provide the kits for the extraction of RNA, especially for either for whole blood or for tissue samples or for culture cells or for body fluids, the kit they provide their instructions where you have to follow all those instructions strictly, and you will get at the end you will get the RNA which is eluted in particular amount of elution buffer. But what is kit based extraction? Kit based, kit means it is a column where you have a bed, and this bed is made up of silica. So silica based, the membrane is silica, which to which your RNA molecules will bind, and after washing that membrane you will get your purified amount of RNA. And the advantage, or you can say that is with the silica based method, is that all the RNA molecule which will bind to this silica, and the whatever flow through is there, this flow through has all the salts or the buffers or the components which inhibit the PCR can be passed through the column. So you have purified RNA which is extracted through the silica based method. But the company people, they say that this silica, they have provided very high salt buffers to lyse the samples, to lyse the cells, to lyse the tissue, so that because of the high salt buffer concentration, less than 200 base pair samples or RNA molecules has low efficiency to bind with this membrane. What does it mean? That means the RNA molecule whose size is more than 200 base pair or nucleotide, it will bind to the silica, and there are less likely chances of binding of these molecules, small RNA molecules, to bind to the silica. That means if you extract your RNA, total RNA, in which, suppose if you are using human sample, so you have 28S, you have 18S, you have 5.8S, you have 5S, and you have tRNA molecules and mRNA molecules, right? So to this silica, the 28S, 18S, 5 and your mRNA, because mRNA are very long molecules in kVs, so these three will bind efficiently compared to 5.8S and 5S and tRNA molecule, because they have less than 200 base pair nucleotides. So these small molecule which are, or small, you can say that mRNAs also, precursor mRNAs or your mature mRNA, mature microRNAs, they will not bind to this particular silica columns, because they have provided in the kit high salt buffers which only allows to bind these large molecules, but the small molecule, this whole small molecules will not bind to the silica to that great extent, so which contribute to 10 to 20% of total RNA. So if you want to extract these molecules like microRNA, if you want to go with the microRNA studies, then you have to have microRNA specialized isolation kits where the microRNA or small molecule will bind to the silica, that silica membrane, right? So with this we can say that you have all large molecule RNases, so RNA molecules in the final elution. So before going to the methodology, which is very, very simple, but we need to understand the concept. So to go into that detail, I want to explain some very, very important points in this particular 10-minute video or 15-minute video that we should keep in mind, because they are very, very important before we are going to start the RNA extraction. The first one is your collection of sample. If I will say to start with whole blood sample, so today we will discuss about totally with the whole blood sample. if you have to isolate RNA from the whole blood, now what you have to see, whole blood has RBCs, WBCs, lymphocytes, and all those molecules, all those cells, right? So when because your RBC are, they don't have RNA, mRNAs, and now you need WBCs, white blood cells, total white blood cells. You now what you have to do, you have to lyse the RBCs. So collection of sample is important, because first you need a vial where your samples, all type of cells you will get intact. So vial where you should not have any clots, so clot free samples you have to collect, like you have to use EDTA vials, heparin vials, or citrate vials. So EDTA vials are most preferred. So if you collect whole blood sample in EDTA vial, if you mix with that, that prevents the coagulation of the blood, and you do not have that clots which will prevent the further lysis, or which further complete lysis, and it will also clog your membrane. So you need clot free whole blood sample, first choice. And the second most important is, the blood sample should be, as soon as you are collecting your whole blood sample, it should be processed within few hours. Like if you keep the blood sample for a long time, the RBC will lyse, WBC will lyse, because they are very fragile. So you have to process the sample within few hours. And if you see the kit literature, most of the kits, they do not say that you can use those kits for frozen samples, right? Remember that point. These kits are not to be used for frozen sample. That means you cannot freeze the whole blood sample, and later on you can isolate the RNA. You will isolate the RNA, that's no problem, but the quality, the quantity of RNA will be suffered when you are doing storing the samples at -80 or -20, or even at any temperature. So second important thing is short lifespan. Some mRNA molecules or some genes which have their mRNA is immediately expressed, and they have very short lifespan. That means they are degraded immediately as we are processing those samples. So because if you are targeting short mRNA which are having short lifespan, that means as soon as you are collecting your sample, you have to process those samples immediately, so that your precious mRNA molecule should not be hampered, or their quality or quantity should not be hampered. So to start, and if we say that, because in gene expression study you will say, okay, I'm comparing the gene expression study with the housekeeping genes. So here I want to tell you that housekeeping genes like 18S, your GAPDH, you are using beta actin. So these molecules are ubiquitously expressed in every cell, and they have huge amount of mRNA copies in the cells. These housekeeping genes, if some amount of degradation is there, even you cannot notice or you cannot see the difference in the gene expression, because they have lots of number of molecules, millions of copies of mRNA present in the cells. And if you process even storing the cells for few hours, even 4 hours, 5 hours or 6 hours, even then your quality of these mRNA will not be hampered. So the housekeeping, you cannot compare your short lifespan mRNA with the short lifespan with the housekeeping gene. So that those 2 points you have to be very careful while you are processing with the or dealing with the whole blood sample. First, collection of sample should be fast. Collection of sample should be in accurate vial, and it should be EDTA or citrate vials or your heparin vials, And because if you're targeting mRNA with a short lifespan, the sample should be processed immediately. And the most important is the kits should not be used for the processing of blood sample which are stored or which are frozen. So you have to go through the kit literature thoroughly, whether they are supporting extraction of RNA from the frozen sample or not. But mostly the kits, they do not allow us to use frozen sample for the extraction of RNA. And if you want to use if you know that you cannot extract RNA on the same day, so what you have to do, there are some kits like PAX gene or PAX RNA systems, where you have to store the samples in these particular vials and then process the vials for the RNA extraction. So these systems are actually developed to where we can store, where you can extract your RNA molecule. The next point, this is short lifespan. The third point is your amount of blood. How much blood you can use. How much blood you can use to get a good amount of RNA. So studies, they show that if you are using 1 to 1.5 ml of blood, whole blood, then you have 4,000 to 7,000 leukocytes. What I said, if you are using 1 to 1.5 ml of blood, then in that blood you have 4,000 to 7,000 leukocyte per ml of your blood. That means you cannot use more than 1.5 ml. No, you can use, but you have to see your application first and how to process the samples. So the kit says you can use 10 to the power 7 cells per spin column. So what I said, yes, you can use more than 1.5 ml of blood, where you will get 10 to the power 7 cells per, you can pass 10 to the power 7 cells per spin column, so that you have sufficient amount of RNA molecule. So if you are using 1 ml of whole blood, you will get approximately 3 microgram of total RNA. How much you will get? 3 microgram of total RNA. So that means to start with 1 ml is okay, because 3 microgram of RNA is sufficient to to go with the study, to go with the some expression analysis or for the PCR. But 3 microgram of RNA you will get. Remember that 3 microgram of total RNA, and whatever amount you are using for the elution. Suppose if you are using 30 microliter elution buffer, then 3 microgram in 30 microliter. If you are using 60 microliter of elution buffer, then you will have 3 microgram of RNA per 60 microliter of reaction. So that calculation part you have to be bear in your mind. So what you will get at the end per microliter, that you have to calculate, and it is very easy to calculate how much you will get. This is approximate value, and this 3 microgram of RNA, it also depends on many factors. So the yield of RNA depends on, first, how much volume of blood you are using. Volume of blood. How much blood you are using for the extraction of RNA. The second thing is the binding capacity of your column. Binding capacity of your column. How much it can bind to the column. So what I said, the yield of RNA depends on, first, amount of blood you are using, second, binding capacity of column, and third one is the time of processing. And the fourth one is the measures you are taking for RNA extraction. What that does mean? Extraction. That means that if you are doing RNA extraction in a very crude manner, then the quality or the amount will hamper. If you know that your hands are full of RNases, your surfaces are full of RNases. RNases are very, very stable enzymes and they can degrade your RNA. Quality of whatever water you are using, whatever buffers they are using, what are tips you are using, how you are using, your lab is clean or not, your apparatuses are clean or not, your centrifuge is clean or not. So all those factors also affect the RNA quality. So, and the time of processing means if you have collected sample in the morning and you are thinking that you will extract RNA in the late evening, that means by that time your total leukocyte count will decreases because they are very fragile, they will break. Third is if you are taking sample in the morning and you are not preserving at particular temperature, at lower temperature, and it is present at the room temperature, and the room temperature is going above 35 degree or 35 degree or 40 degree or close to 42 degree, then this will affect your yield of the RNA. So the second point is binding capacity of the column. So the kit literature says that mostly in most of the kits 100 micrograms, what kit says, 100 microgram of nucleic acid. How much? 100 microgram of nucleic acid that can bind to the silica column. But it also depends whether your cells are completely lysed, one thing. Third thing, your sample is already lysed and your RNA is degraded, then the capacity also decreases. Third is if your lysis buffer is inappropriate to the sample volume. Like suppose if I will say you have to use 3 volumes of lysis buffer to the sample volume. That means if you are using 200 microliter of your sample, that means you have to use 600 microliter of lysis buffer, and you are using just double or just same volume. That means that is inappropriate to get sufficient amount of RNA at the end. So and the amount of blood, if you are using more blood, if you say I will use 5 ml of blood, so you know that the binding capacity of the column is 100 microgram only and you are using more blood. So that means you are wasting and you are inhibiting your binding capacity also, and you are clogging your membrane also, and because of the high amount it is insufficient like space or reactions to be that interaction is not proper in the of the nucleic acid with the silica columns. So all these points are very, very important, and the most important point is this is the amount of blood. The fourth point is amount of RBCs. You know that RBCs, they don't have mRNAs, active mRNAs. So you don't need RBCs to be lysed. So what you know that 1000 times more number of RBCs than your WBCs or leukocytes. So but you don't need this particular population of cells, you need this group because it has expression, it has RNAs. So RBC, you have to get rid of RBCs, and in the RNA extraction, kit based RNA extraction, they are providing RBC lysis buffer. You know that if you are lysing your RBC, okay, right, but your lysis is not complete. That means that will contaminate your WBCs, and if your WBCs are not, if you are not getting pure population of WBCs, the RBCs, they will also, because of hemoglobin and many other contaminants are there, it will inhibit your further PCRs or reactions. So first you have to get rid of RBCs by adding RBC lysis buffer. And if your that WBC cell population is not clear, if you are not getting clean WBC cell population, till that end you have to wash the cells with the RBC lysis buffer to get rid of all the RBCs, right? So, and people, what they used to do, the another way of this is by using lysis buffer. People, what they will do, they isolate peripheral blood mononuclear cells, PBMCs, by Ficol gradient. As you know that this is very common method. So they used to go with the PBMC isolation method. So both method are important, both are right and easy method. You can go with any method, right? So now the fifth point is, the important point is, fifth is leukocyte lysis. Now once you have removed all the RBCs, you have palette of leukocyte. Now you have to lyse this palette, because this is your interest because these contain these cells, they contain your target genes or gene of interest. So whenever you have to lyse the leukocytes, you need to ensure that the lysis should be complete, and for that the kits, they are providing shredder columns. One. Second one, there is a homogenizer method, and third one is rotors and pestles. So to ensure the complete lysis of these cells, leukocytes, there are shredder columns. There is another way to ensure the complete lysis, that is using homogenizers, and the third one is using rotor and pestles. So rotor based homogenizers are not preferred for the cells, they are preferred for the tissues. Homogenizers are also preferred for the tissues which are hard, where we cannot easily lyse the cell, so you have to use homogenizers. Either they are manual homogenizer and they are motor based homogenizers, both you can use. Third one is shredder columns. Mostly kits, they provide shredder columns where the lysis is achieved by these columns. So before going to detail in the leukocyte lysis, one very important thing what you have to remember is that you have to use BME, beta mercaptoethanol, or DTT, diethiotriatol. So these molecules, these are the denaturing agents, and they help in the lysis of the cells, complete lysis of the cell. So your leukocyte lysis buffer should contain these two, either BME or DTT. That I will explain in the material method section. So these are some instructions while you are using kit based RNA extraction. So to end with this lecture, this is the instructions. You just follow these instructions, write down and pen down and noted down these instructions, because these are very important points. So thank you, and in next lecture what we'll learn the procedure of kit based RNA extraction, and how to prevent the contamination, and how to decontaminate, or how to prevent the contamination of your RNA or degradation of your RNA, and how to prevent degradation of RNA from your RNases, and what you can measure you can take before going to the RNA extraction. So that we'll learn in the next lecture. Thank you.